so what we're going to do is we're still in chapter 14 and we're picking up kind of the middle third-ish of the textbook, or chapter content, and we're talking about resistance factors to the flow of blood. I mentioned a moment ago that resistance factors have an inverse relationship to blood flow. And I said, well, we're going to talk about three things that actually influence resistance. So that's where we're at. The three things that influence resistance to the flow of blood through the body. The first resistance factor is called blood viscosity. What does viscosity mean? Thickness. The blood thickness or stickiness of the blood is considered a resistance factor. The more thick blood may be, what do you think the relationship? If our viscosity goes up, our blood flow must go down or decrease. So the relationship is inverse one another. The relationship between blood viscosity and blood flow is inverse. The more viscous a fluid becomes, the less it will flow, the slower the rate it will flow. I have a note here that it's relatively constant. What I mean is it's not going to fluctuate from millisecond to millisecond, like say your polarity of a nerve cell would. So right now your blood has a certain viscosity, right now it has the same viscosity, right? And so we can measure it over a period of a day or something, it might change. But it's relatively constant. It's not going to fluctuate quick to have really major impact on the distribution of blood through a given organ at any given time. Okay? So your blood, right, depending on your hydration, hopefully it's in set point, right? And so that's going to be relatively consistent. Depending upon what you eat and drink, or if you're diabetic, for example, or have other types of blood diseases, for example. So certainly hydration, dehydration can influence blood viscosity. If you're an athlete and you go train in Denver, because it's considered the Mile High City, and you increase your blood count, okay, or you dope blood and you add blood, your blood becomes more viscous. And so there's many, you could influence your blood viscosity outside of just diet as well. You could actually inject blood into your body, red blood cells, or you could eat differently, or if you eat an unhealthy meal, like okay, so what's the Oklahoma or state food? Is it chicken fried steak, right? So you're good Okie, you go eat a good old chicken fried steak, and what's going to be in there besides all that yumminess? What's in there? What is that yumminess? Lots of good old fats. And so you could do a blood draw. I've seen people do blood draws after eating a fat, like going to even IHOP and eating some pancakes. And there is a measurable, like 2-3 centimeter measurable bit of just fat. So that is certainly going to influence the viscosity. And so after eating a meal like that, you're probably not going to want to exercise. And if you did, yeah, you're in trouble. But also your blood flow is distributed differently. But it will certainly influence the viscosity. So it is described as being relatively constant, but what you do, just like anything else, will influence it. Another resistance factor is called the blood vessel length. The length of blood vessels are not all the same. Some are short, some are long. However, in an adult, the blood vessel length should be relatively constant. And you shouldn't see, in this millisecond, a blood vessel being a meter long, and the next minute, a centimeter long, right? It's going to be pretty consistent throughout your life. But through different organs, right, blood vessels have different lengths. What is the relationship between blood vessel length and resistance to the flow of blood? So what's the correlation?
I think the longer it would take is slow it down?
Correct.
Because it has more surface tension, I guess it has to go over.
More surface that it has to drag along. So if the blood vessel length goes up, so does the resistance increases. So if I increase the resistance, the blood flow then will be decreased through longer blood vessels. So the blood vessel length, longer blood vessels contribute a greater resistance. So the relationship between length and blood flow is inverse. This is a resistance factor. A third resistance factor to the flow of blood is the diameter of the blood vessels, which we've been using this example honestly all semester long when we did it, even back when we did our first lab experiment, when we did the temperature regulation experiment we talked about. We introduced vasoconstriction and vasodilation there. The blood vessel diameter can change quickly if your environmental factors change or your nervous system decides to change the resistance factors pretty quickly. Because we can quickly change the diameter, but cannot quickly change the viscosity or the length of a blood vessel, if you want to change the blood flow through a particular organ, what do you need to change? The diameter of a blood vessel. This is going to have a quick impact on my abbreviation. The PR stands for peripheral resistance. It's going to significantly influence the peripheral resistance, to the flow of blood. So what I want to do is turn to figure 1410 to illustrate how the blood vessel diameter can change, reinforcing some terms we learned way back in August. Looking at 1410, blood vessels are either relaxed or constricting or fully dilating. Okay? In this image, we're looking at arterials. Now I want you to turn your attention to reminding you that the arterials do have smooth musculature. In the first image, we see it under just kind of normal conditions. Notice the diameter of the lumen. The next scenario, what do you think the muscle has done? It is constricted. And what has happened to the diameter? It's getting smaller. And so the lumen has gotten smaller. Our resistance factors have increased. And so downstream to some capillary, the blood flow must, if this is leading up to a capillary, the downstream blood flow must decrease. If we increase that resistance, the blood flow to the next blood vessels is going to decrease. So you might see, for example, vasoconstriction to arterioles of your skeletal muscles when you're taking a nap. You don't need excessive blood flow to your skeletal muscles. They just need enough. You have to kind of keep them at rest. However, if you get up and start moving, your blood vessels are going to need more blood, so the arterioles leading up to those muscles must dilate. To do what with the resistance? Decrease the resistance and speed up the velocity, the flow of the blood to that downstream vessels, downstream organ capillaries. So the purpose of this is to illustrate is that the bulk of the distribution of the blood through our body is dependent upon arterioles. This is reinforcing what we're studying in lab this week about the afferent arterioles that take blood through your what? Glomerulus. To do filtration in your kidneys. So same concept. Here we're just talking about any arterial throughout the body.
Can I please ask a quick question about the length again? You said inverse relationship, but for some reason I have increased length equals increased resistance, but you're asking for the relationship for blood flow.
Yeah. So if you increase resistance, the relationship to, I could have said it two ways. It's a direct causative for resistance, but if my resistance goes up,
Okay.
the blood flow would go down. And so there's two different ways to look at it, blood flow or resistance. Did I answer?
Yeah, so it's, I mean, it's just 3 steps in this one.
Longer blood vessels have more resistance, so compared to a shorter blood vessel, it would take longer to get the blood.
Yeah.
Longer for the blood to get to a longer blood vessel.
Just want to make sure which of these 3 steps.
You're okay. Thank you. Your body is going to autonomically control arterial constriction and dilation for most blood vessels throughout the body. Some are intrinsically controlled, like at the kidney. But your body's going to match blood flow with tissue demands. So the scenario I gave, if you're resting, you want to have minimal blood flow to an inactive organ. If you're exercising, you would want to increase blood flow to, say, your skeletal muscles, but decrease it to, say, the digestive organs, for example. So we got to match it. And so in the first scenario, I don't remember what the colors are. So under normal resting conditions, we have some respiratory gases. So some tissue, let's say this is your skeletal muscle tissue, for example, at rest. They're going to take in oxygen and they're going to give off carbon dioxide. Okay, it's going to match. I want you to look at the arterial in the diameter. If the cells become more active, which gas are they going to produce more of? They're going to produce more carbon dioxide. Okay. That carbon dioxide, we know, can influence the pH, and so that's going to start to locally change conditions. So what should the blood vessel do in this tissue that's producing more carbon dioxide? That blood vessel should probably, which one? Dilate. It should dilate. When it dilates, we can pick up more of that excessive carbon dioxide and bring the tissues back to set point. Okay? And then at some point, right, the cells are more active. We pick up more carbon dioxide. That increased blood flow brings in now what? We're really just trying to pick up the extra carbon dioxide, but we increase the blood flow to pick it up. And now we can also deliver more oxygen to match the increase. What does this stand for? Metabolic rate. And so at some point, we're going to slow down the activity. So what should this arterial do? Constrict. Your body's just going to precisely control vasoconstriction and vasodilation to match each tissue needs. We okay with that? Homeostasis. Go ahead. All right. Figure 14.14 illustrates this as well. It's talking about the myogenic response in perfusion pressure. So let's just work through this title first. Pressure we can handle, right? Perfusion, blood flow pressure, changes that cause a response by the muscle, the generating muscle. Your blood vessels have muscles in them, and they can stretch if your blood pressure goes up, and they will probably recoil if the blood pressure goes down. And so we have sensors. And we talk about everything has a set point, and so your smooth muscle arterial the smooth muscle in your arterioles also have a kind of a set point for their stretching or recoiling. Or we could say myogenic response. What are we looking at here? This is in a scenario if blood flow increases to a particular organ, moving the wrong way. The pressure of blood flow through a particular arterial will read this image correctly. If you're looking at blood flow in an arterial, the blood flow goes up, okay, causes the increase An arterial is going to do what? Stretch. And so there's going to be more blood flow into that particular vessel. That causes the smooth muscle of the arterioles to stretch out, which then initiates them to, so they stretch, and they're going to respond to that stretch, and now they're going to kind of constrict a little bit to increase resistance to bring the blood flow back to set point. And this is all another example of negative feedback. And so another maybe way to illustrate this is to say that some smooth muscles are intrinsically regulated based upon the blood flow. Some arterioles are intrinsically regulated, or we could say myogenic response happens. To make sure the blood flow to a particular organ matches the demands of that particular organ. So some smooth muscles in arterioles are intrinsically controlled. And then a side note, they're also controlled how? Extrinsically by the what can extrinsically control my smooth muscle? The what? What can control my smooth muscle? We had a test over it. Autonomic nervous system. But if we want to do it even faster than autonomic nervous system, we need to have a source of. If I want to control something faster, I need a source of intrinsic control. Autonomic nervous system's fast, but if it's controlled right there, that's even faster. And the way that these some of these arterioles work is they just respond to stretch. And this is showing that negative feedback. If they stretch too much, brings too much blood flow, so they need to constrict to decrease the blood flow back to set point. If you need to make a reference to figure 14. As I mentioned, your body is going to precisely moderate that blood flow. You know that you can quickly change from resting to an excited, and then maybe take a while to get from excited to a resting state. And so as your tissues become more active, we need to increase blood flow to those tissues to keep us in homeostasis. And figure exercise 14.1 is illustrating the regulation of blood flow and where it goes in a resting state or under parasympathetic control versus an exercising state, so under sympathetic control. You notice that under rest, the bulk of your blood flow gets distributed where? Get a lot to the GI tract, a lot to your kidneys, skeletal muscle, brain, and everything else. Okay, so we're looking at a liter for the skeletal muscles per minute. Kidneys also liter per minute. The GI tract 1.25 liters, and the brain 0.7. As you transition to an exercise state, the brain, even though it's not correspondingly increased, we're still getting about the same amount of blood flow to the brain at about 0.8 liters per minute. So the brain is still important during exercise, okay? Your skin blood distribution actually goes what? Up, because we need to get rid of heat. We saw that, and we measured that in that first lab that we did. We saw the blood flow change there. The heart went from 0.2 liters up to 1.15, so we see a significant spike here. The GI tract see a little bit of a decrease, but we don't stop blood flow to the digestive system. We don't want to kill it off. We just want to slow down this metabolic activity. But however, we see a significant increase in cardiac output to the skeletal muscle, 20-fold in this case. You didn't increase your blood volume, you just increased your what? Cardiac output significantly under exercise. And so the way the body distributes this is just increasing arterial diameter for these various organs and increase our heart rate and stroke volume. And if we slow down, everything will go back to that set point distribution. So the summary of this is, one, not all organs receive the exact same amount of blood flow, and then two, it can be quickly changed as we go from a resting to an exercising state. Table 14.2 should be redundant as well to what we've been talking about this semester, in that not only can we intrinsically control the arterial diameter, but we can extrinsically control it by our autonomic nervous system. Highlighted here is the sympathetic nervous system. Can cause some blood vessels to constrict. And I don't think this is an all-inclusive list. I think, this is not all-inclusive. I don't know why. I don't know there's quite a bit of information missing here, but kind of getting an idea that the sympathetic nervous system can extrinsically control arterial diameter. So can what? Hormones. Makes sense. Okay, tying in where we've been. Autonomic the nervous system can override things extrinsically, and hormonal factors can override, so including constriction. What else could be added here? What else could we add? Besides nerves and hormones, we could add blood factors, or we could say what? What word have we been using? Humorally. So, not all blood vessels are intrinsically regulated. They're also extrinsically regulated by the autonomic nervous system, hormonally, and humorally. That should be a redundant theme right up till this point by now. I'm going to turn our attention now to our capillaries. We introduced these in chapter 9. We talked about the blood-brain barrier. So these images should look kind of familiar. If you recall, we said our capillaries throughout the bulk of the body are very porous. Or these paracellular pathways exist, or analogous. They're not gap junctions, but they're physical pores between the membranes of cells of the capillaries. The purpose of what is the purpose of these pathways? To what? To allow exchange, to allow for bulk flow. Okay? Some particles can easily just diffuse through these spaces. Some capillaries are called continuous capillaries. And then I want to turn our attention to another type of capillary called a fenestrated capillary. What can you tell about the difference between a fenestrated versus a continuous capillary? The pores
are not there on the continuous capillary.
They're there in the continuous capillary, but much smaller. So there's still a pathway, but it's just significantly smaller. So your continuous capillaries are less porous than the fenestrated capillaries. Either way, the purpose of the capillaries is to carry out bulk flow. Okay, so if we turn our attention to how a capillary works, this image should look familiar from chapter 9, blood-brain barrier discussion. If we want to push, right, or filter something from the plasma to the interstitial fluid, we've got to go back to what we studied in chapter 4, the size and the solubility of the particle. If something's too big, is it going to filter out? No. If it's too small, could it filter? If it's so small, could it filter? Yes. If something's lipid soluble, could it filter? Yes. If something's water soluble and giant, can it filter? No. Unless we have a form of what? How do I carry proteins across cells? Vesicles, if I vesiculate them. So sure, some proteins could, but we've got to intentionally endocytose them, traffic them, and exocytose them. So this is going to summarize that size and solubility for filtration. Furthermore, if you've got your guided notes, I want you to draw a giant red blood cell in here. So draw the entire space. Draw a big giant red blood cell back in the background. Our red blood cells are as wide or if not wider than your capillaries. So they're jamming in there. They're squeezing into your capillaries. So can our red blood cells filter into the interstitial fluid? No. But more lipid-soluble substances like what? What's a small lipid-soluble that could easily filter? Oxygen? Potassium is charged, so it's more not lipid-soluble, it's more water-soluble. So it could not diffuse across the cell, but it could diffuse what? Between. So through the little pores. There we go. So what else besides oxygen is probably small lipid-soluble that could simply diffuse? CO2 and hormones, right? And what kind of hormones? Steroid hormones. So not all hormones can simply diffuse, like say, because they're good for an insulin, that's a little protein. What is going to push out? What's going to help push? A gradient, what kind? Pressure gradient. And honestly, there are other gradients, but we're going to put our blinders on the pressure gradient. Pressure is going to push lipid-soluble substances to do what? What kind of process is this? Chapter 4 review. Lipid-soluble substances can simply diffuse. Through cells. More water-soluble substances, such as our ions or small proteins, can also diffuse, but they have to go where? Through the pores. So sodium, potassium, glucose, etc., they can diffuse, but not through the cells, but through the pores. The pores are big enough to filter and reabsorb these substances. Larger proteins that are too big to diffuse through the pores have to be what? Transcytosed. That means we have to endocytose them, traffic them, and then exocytose them. But if a protein is too big to diffuse through a pore, doesn't have the ability to transcytose, where is it? What happens? It stays in the capillary. If the pressure is pushing out, what can we label on here? Pressure is pushing out. Filtration, so label each filtration. And also reabsorption can happen. And so bulk flow uses simple diffusion. Diffusion through pores, transcytosis, to move different sized particles and different soluble particles between the blood plasma and the interstitial fluid. The This is figure 1418. It's a culmination of really chapter 14, or excuse me, chapter 4 information and the introduction to capillaries in chapter 9. With that, I'm going to go ahead and transition into the next image, which is figure 1419. Your textbook goes into quantifications, which I'm not going to hold you guys accountable for. We've been using the word pressure grading. We haven't been calculating a lot of things in this class. But if I want to filter something, the pressure should be higher where? In the capillary than in the what? Interstitial fluid. And if I want to reabsorb something, the pressure should be higher in the interstitial fluid than in the capillary. That's what this is quantitating, filtration versus reabsorption. Then we will use this abbreviation though, I think in the renal system at the very end, NFP stands for net filtration pressure. So go ahead and write down that abbreviation, net filtration pressure. That tells us there's so many influences on pressure and filtration. But what we want to focus on, if we want to push out, we want to filter, the pressure needs to be higher where? Inside. And if we want to reabsorb, the pressure should be higher in the interstitial fluid. And so this is showing the quantification. And the arrows show you the net direction. Capillaries carry out both filtration and absorption in exchange. We tend to see the bulk of filtration happening near the what? Arterial, where that blood's coming in at a higher pressure, and more reabsorption occurring towards the venial end. But both occur across the capillaries. It does say 38, that is tiny, and the net reabsorption is 26. Again, I'm not going to hold you accountable, but your book has the values, so I just went ahead and showed them. So then the takeaway is the filtration pressure is higher than the absorption. By the time, right, so if we put ourselves into perspective, we're looking at a capillary in your body, velocity has declined, pressure is declining, and then the velocity is going to pick up as the pressure continues to decline. And we need to get blood from the most distant part of your body back over to the right side of your heart. Pressure is low, velocity is still kind of low, not as low as it was in the capillaries, but gravity pulling, right, you may be laying down, whatever, we need some help getting that venous blood back to the heart. So we know that there are valves in the veins, but we also need to move. If you want blood flow returned to the heart, you need to move. And by use of this thing it's collectively called the skeletal muscle pump. You only have one heart, but your skeletal muscles help squeeze on venous blood to help it propel back to the heart. So that's why if you work at a medical facility, you've got someone who's bedbound, just had surgery, they may have some physical therapy for just massaging or hook them up to a lot of times we hook them up to the machines anymore, like the compression machines, for example. Even if they don't have a leg injury or something like that, you'll see these devices put on for blood flow. You want to get the blood flowing to help. There's many benefits besides just returning blood to the heart why you want to have increased blood flow. But so moving helps return blood back to the heart. When you physically move a muscle, you recall we said those muscles squeeze on your blood vessels, not only the arteries, but also the veins. And when they squeeze, it gets a little extra propulsion. And the blood will go back to the heart, not away from the heart because of our valves. And so this is the action of this is called your skeletal muscle pump. There is another pump called the respiratory pump. I don't know that I have it shown here. I don't have it shown. There's another pump. We'll go ahead and make a side note. There's also a respiratory pump. respiratory pump. When you breathe, time back to what we learned in the previous lab, you create a what? Pressure gradient. Same concept. When you breathe and change the pressure in your thoracic cavity, it alternates with the abdominal cavity. So these breaths, when the pressure is high in your abdominal cavity, it pushes on the veins and helps push blood back to the heart. So breathing helps return blood back to the heart. And what is this referred to as? The respiratory pump. I want to spend the next 2 minutes to just introduce the lymphatics, okay, and then we'll conclude class in just a minute. We've not talked about the lymphatics, but the lymphatics are also another set of vessels. They don't carry blood, but they carry what? What do they carry? What do we call it? Call lymph. Your lymphatic vessels carry lymph. If you recall about the capillaries, the inward absorption pressure is lower than the filtration. Additionally, if proteins like hormones are made by tissues, you might have trouble absorbing them into the blood. And so what happens is you have to have an accessory route to get large proteins or fat from our diet back into the circulatory system. And so your lymphatic vessels are this initial way that interstitial fluid, large proteins and fat globules, can have a route to get into the circulatory system. So your lymphatic vessels pick up interstitial fluid that has not been what? It hasn't been reabsorbed by your capillaries. It starts at where? They start at your capillaries. And the fluid goes through different nodes, which you've probably heard of before, for an immune check. It's like a security station, pretty much. And then returns back to where? Your lymphatic vessels drain into the vena cava. And so anything picked up by the lymph is going to go straight into your vena cava, straight into your blood. We will conclude our discussion here.